Hello, today we are going to explore that how enzymes work. By the end of this lesson, you will be able to explain the role of active site, formation of enzymes and substrate complex, and how enzymes lowers the activation energy, enzyme specificity, and you will be able to compare the models like induced fit model and lock and key model. So let's start with a simple question that how can an enzyme speed up the chemical reaction without being used up? To answer that, we need to understand that what is enzyme, how the structure of enzymes actually allows to perform function. So enzymes are actually a globular proteins. They act as a catalyst. They called as a biological catalyst. A catalyst are the ones that can increase the speed of reaction without being used up. So they remain unchanged. There are two types of enzymes, intracellular and extracellular enzymes. Intracellular enzymes, the words indicate that intra, inside the cell. These are the enzymes that work inside the cell, while extracellular enzymes, extra mean outside. Thus, enzymes that work or catalyze the chemical reaction outside the cell. So, the key question is now, how does enzyme actually catalyze the reaction? They are catalyzing the reactions, but how does? This is the key question. For that, look at this picture. Here you can see this is an enzyme, and notice that there is a specific region. This specific region is called active site. You can see that this active site has a specific shape. Because of this specific shape, this active site allows the substrate to bind with it. And the molecule that bind with it is called substrate. Look, this is the substrate. The shape of the substrate is exactly specific with the enzyme. As you can see, the active site is very specific. An enzyme does not normally interact with every molecule around it. That's why enzymes are called as specific. This is known as enzymes specificity. For example, an enzyme that catalyzes the breakdown of particular substrate will have the active site that is complementary to the substrate. This is the enzyme and this is the active site. This active site is complementary to the substrate. If the shape of active site changes, what do you think will happen to enzyme activity? Exactly. If the active site's shape this is altered, the substrate may no longer be affectedly bind to the active site. Now, look at what happens when the enzyme and substrate combine with each other or approaches each other. The substrate will bind to the enzyme, the on active site. And at this point, we have an enzyme substrate complex. We can represent the process as enzyme E plus this substrate S into enzyme substrate complex. And this one is enzyme substrate complex. The substrate will be converted into products during the reaction. These are products. And the enzyme substrate complex will change after the reaction into products. And these are products. Importantly, you can see that the enzyme itself is not permanently changed and can therefore participate in another reaction. But we still have not answered a very important question, that why does binding the substrate make the reaction faster? after binding the substrate with enzyme, why the reaction rate becomes faster. That brings us the activation energy. Every reaction requires a certain amount of minimum energy to begin the reaction. This is called activation energy. Here, without enzyme, this is the total amount of energy that is needed to start the reaction. Suppose this is the chemical reaction and without enzyme, it need this much amount of energy supplied to start the reaction. This is called activation energy. And this one is lower curve. This represents the reaction with enzyme. Notice something very important. The enzyme provides an alternative reaction pathway. You can say this reaction pathway will be starting from the same point, but the pathway is different from that with no enzyme. Therefore, the substrate with having an enzyme have enough energy to complete the reaction faster and to move the reaction with a faster speed as compared to the substrate without enzymes. It needs more energy to react and then successfully complete the reaction. So remember, enzymes increase the reaction rate by lowering the activation energy. They do not provide energy for the reaction. They do not change the overall energy difference between reactants and products. They actually increase the reaction rate by decreasing the activation energy. All energy released during the reaction will always be same with enzyme or without enzyme. The overall reaction energy or the energy released will be the same. So now let's consider how the substrate bind to the active site. We will discuss two models. First is lock and key model. Imagine a lock and a key. As a particular key always fits to a particular lock because of their shape, which are complementary to the key, in a same way, according to lock and key model, the enzyme's active site is very specific and has a specific shape that it is complementary to the substrate. The specific substrate will bind to the active site of the enzyme. The reaction will happen. This model helps us to understand that enzymes are very specific in their actions, in the same way like lock and key. But there is a limitation about this model. The active site is not complementary rigid. This leads us to the second model, that is induced-fit model. Induced-fit models actually provide a more accurate explanation of enzyme action. According to this model, the active site is not perfectly rigid structure. And when the substrate approaches to the active site of the enzyme to bind with the enzyme to perform the action, interactions between substrate and enzyme can cause the active site to change slightly and the active site become more suitable or complementary to the substrate. Here you can see enzyme substrate complex and the active site is slightly changed. This is called induced fit model. The difference between lock and key model and induced fit model is this. In lock and key model, the active site is you can say considered as already have a complementary with the substrate. This is complementary with the substrate. And in induced fit model, the binding of the substrate cause the active site to change slightly to provide the better fit. So, how an enzyme catalyzes a reaction? Should include some specific key points. For example, first, you will explain the active site. You will explain that for the enzyme to catalyze the reaction, First, it need to bind with the active site. Active site is very specific to the substrate. This forms an enzyme-substrate complex. Because of the enzyme, it provides an alternative pathway with lower activation energy, and this will increase the rate of chemical reaction. Enzyme is out without permanently changed, and it can be used again and again, and our product will be formed. So now that we have understood what is enzyme, what is the mode of action, the next question that we will cover tomorrow is which factors can affect the rate of enzyme, like temperature, pH, enzyme concentration, substrate concentration, and inhibitors.